Hello everyone, my name is Min-Soo Choi. I'm in a laboratory for systems biology and bio-inspired engineering. I will give the lecture of front part of chapter 11, Therapeutic Hormone. First, I will introduce you about the general definition of hormone. Then we will discuss three endocrine hormones, insulin, glucagon, and growth hormone for detail. Originally, the term hormone was defined as a substance synthesized and released from a special gland in the body to generate a change in that target cell by interacting with a receptor. Hormones travel to the target cell via the circulatory system. In this lecture, we will introduce three endocrine hormones including insulin, glucagon, and growth hormone. As you can see left figure, insulin is polypeptide hormone produced by the beta cells of pancreatic islets of Langerhans. Its most important targets are skeletal muscle fibers and hepatocytes and adipocytes. It plays a central role in regulating blood glucose levels. Insulin orchestrates a suitable metabolic response to the absorption of glucose and other nutrients in a number of ways. It stimulates glucose transport into cells and reducing their blood concentration, and intracellular anabolics such as glucagon synthesis. Furthermore, insulin inhibits catabolic pathways such as glycogenolysis. Failure of the body to synthesize sufficient insulin results in the development of insulin-dependent diabetes mellitus. This is also known as type 1 diabetes. This picture describes a contrasting situation in a normal and IDDM condition. IDDM is caused by T-cell mediated autoimmune destruction of the insulin-producing beta pancreatic islet cells in genetically predisposed individuals. Insulin was first identified as an anti-diabetic factor in 1921 and was introduced clinically the following year. Pre-proinsulin is transcribed as a 110 amino acid chain, and proinsulin is produced by the removal of the signal peptide. Proinsulin is proteolytically processed in the coated secretory granules, yielding mature insulin and 34 amino acid connecting peptide, C-peptide. Removal of the connecting C-peptide chain produces mature insulin. The C-peptide is further proteolytically modified by removal of a dipeptide from each of its ends. The insulin is stored in the form of characteristic zinc insulin hexamer, consisting of 6 molecules of insulin stabilized by 2 zinc atoms. Major insulin consists of 2 polypeptide chains connected by 2 interchain disulfide linkage. The A chain contains 21 amino acids, whereas the larger B chain is composed of 30 residues. The insulin receptor has 2 alpha subunits and 2 beta subunits. These are held together by disulfide linkage. Binding of insulin to its receptor promotes the autophosphorylation of these 3 specific tyrosine residues in the tyrosine kinase domain. Activation of the beta subunits tyrosine kinase activity results in the phosphorylation of various intracellular substrates, which trigger the MAPK and PI3K kinase pathway responsible for inducing insulin's mitogenic and metabolic effects. This table shows the list of insulin-sensitive genes. Traditionally, commercial insulin preparations were produced by the direct extraction from pancreatic tissue of slaughterhouse pigs and cattle. However, the use of animal-derived products had a number of potential disadvantages, such as immunogenicity and availability. These issues and concerns result in the development of recombinant human insulin products, now routinely used in the management of diabetes. The initial approach to recombinant insulin production taken entailed inserting the nucleotide sequence coding for insulin A and B chains into two different E. coli cells. It was termed human insulin (CRB). An alternative method entails inserting a nucleotide sequence coding for human proinsulin into recombinant E. coli. This approach has become more popular due to the requirement for a single fermentation and subsequent purification scheme. Such preparation have been termed human insulin (PRB). A right picture shows a purification scheme for human insulin (PRB). A final RP-HPLC policing step is a highly pure product of insulin. Insulin may be formulated in a number of ways, generally in order to alter its pharmacokinetic profile. This graph shows subtypes and approximate duration of actions of different insulin formulations. Rapid-acting insulins are those preparations that yield an elevated blood insulin concentration relatively quickly after their administration. On the other hand, long-acting insulins enter the circulation much more slowly from the depot site. Therapy consists of injection of slow and fast-acting insulins or a mixture of both. In order to prolong the duration of insulin action, soluble insulin may be formulated to generate insulin suspensions. This is generally achieved in one of two ways: addition of zinc or protein such as proteamine. Recombinant DNA technology facilitates native and engineered human insulin products. By generating the engineered insulin, it is possible to make faster acting, slow acting insulins or super potent insulin forms. This table is about the native and engineered human insulin preparations that have gained approval of general medical use. There have been attempts to improve upon the mode of administering insulin, as many people find injection inconvenient and generally painful. Insulin is usually taken as subcutaneous injections. There is an alternative means like inhalable insulin. Open loop system consists of an infusion pump. Blood glucose levels are monitored manually and the infusion rate is programmed accordingly. They have not as yet become popular in practice, mainly due to inconvenience. The closed loop system, it is often termed the artificial pancreas. It consists of pump, infusion device, an integral glucose sensor, and computer. The system imitates the feedback loop inherent to the pancreatic beta cells to control glucose levels. Furthermore, transplantation of insulin-producing pancreatic cells is being investigated. The left picture shows the intimate relationship both insulin and glucagon have to each other. The major biological actions of glucagon tend to oppose those of insulin. When blood glucose concentration decreases, insulin levels decrease, and the catabolic effects of glucagon become more prominent. The prominent metabolic effect is to increase blood glucose level. Glucagon initiates its metabolic effects by binding to specific cell surface receptor. Then a membrane-bound adenylate kinase is activated. This promotes activation of cyclic adenosine monophosphate (CAMP) dependent protein kinase. This kinase phosphorylates key regulatory enzymes in carbohydrate metabolism. As you can see, growth hormone is an anabolic hormone made and secreted by the pituitary gland. Growth hormone is a large polypeptide thought to encourage growth indirectly by stimulating the release of growth hormone factors from the liver and muscle. These growth factors create a cascade of events typically associated with higher growth hormone concentrations. Growth hormone is released in response to growth hormone releasing hormone produced by the hypothalamus. Growth hormone primarily displays an anabolic activity. It partially stimulates the growth of bone, muscle, and cartilage cells directly. During the years of active body growth, deficiency in the secretion of human growth hormone results in pituitary dwarfism. On the other hand, overproduction of human growth hormone results in giantism. After primary body growth, human growth hormone overproduction has occurred, resulting in acromegaly. As you can see, the left table, growth hormone has a potentially wide range of therapeutic uses. Growth hormone is now produced by recombinant DNA technology and is prescribed for a variety of reasons. The right table shows recombinant human growth hormone preparations approved for general medical use. Thank you for attention.